Well, hey, AP, so here is the Section 3 notes 5.3. And so what you can see here is we're talking about active transport, which requires energy. And so this is going from low to high concentration. So we're going uphill, okay? That's where we're going, from low to high. It requires energy, and usually it's in the form of ATP. Okay, so here's some examples of moving glucose and sodium, but the biggest example that we're going to go through, this an illustrative example in biology, is the sodium-potassium pump. So the sodium-potassium pump is this really important pump that pumps sodium and potassium against their concentration gradients. Okay, so what I want to show you is this. What you have here, here's the pump, and you have, notice the starting situation. You're going from high sodium outside, you have low sodium inside, and you have low potassium outside and high potassium outside, okay? And so we're going against both concentration gradients of ions. So you bind to 3 sodiums. Here's ATP. ATP is being used. The protein breaks down ATP, hydrolyzes it, and then you release the three positive ions outside the cell. Notice how the sites now are open for potassium. Potassium binds, pumps it inside the cell, and so you're pumping 2 out. And so you're pumping 3 positives out and 2 positives in. And if you do this thousands and thousands of times with lots of different proteins, you're creating kind of like a battery. You have a positive charge on the outside and almost kind of like a neutral charge or a very different charge on the inside of the cell. And this allows cells to respond to the environment. Okay, very important. And so how important is it? Well, there's actually a poison called ouabain, and this poison shuts down the sodium-potassium pump, and it leads to death and paralysis. Here is just kind of the sodium-potassium pump in an animation. So lots of different reasons why the sodium-potassium pump is important. Would be cool to kind of look this up and figure that out. And so here's membrane transport. Simple diffusion, no energy is required. So this is a helpful table. A concentration gradient. Facilitated diffusion needs a protein. There is specificity, which means we're looking for specific molecules. And then active transport, of course, requires energy. ATP hydrolysis is breaking down ATP, and when we do that, it's a conformational change. Okay, there's 2 types of active transport. There's primary active transport, which is directly using ATP, and then secondary active transport uses the energy from an ion concentration gradient that was set up by primary active transport. Okay, so it's set up by this. The best way to learn this is an example here in a second. So membrane potential is the voltage. So the sodium-potassium pump actually creates this membrane potential. Why is membrane potential important? Well, there's your EKG for our heart. You also respond, your nervous system responds because there's a membrane potential. And so how do you know like an ant or a spider is crawling up your leg? Well, they have to send a signal. That signal is sent because of membrane potential. There's like a charge. We're running a small voltage. Okay, so voltage is created by differences. And so what happens is a channel is open. All those sodium ions rush into the cell, and we have depolarization and repolarization, which you'll learn in anatomy class, and we allow the heartbeats to beat and the body to respond. Okay, 2 combined forces collectively called the electrochemical gradient drive the diffusions across and allows us to respond to environmental stimulus and beats. Chemical, of course, is ions, and electrical is the effect of membrane potential because they're positively charged. Okay, and so here's a proton pump. So the sodium-potassium pump is a major electrogenic pump, which means it generates voltage. Yes, you do have a voltage. But the main electrogenic pump of plants and fungi and bacteria is called a proton pump. It's different, but similar. So here's a proton pump. We use an ATP, but instead of like sodium and potassium, we're just pumping hydrogen ions, aka protons, across the membrane. And again, we're just kind of setting up a voltage. Positive on the outside, neutral and negative on the inside by comparison, and we have a voltage, okay? Co-transport occurs when a transport protein can couple is the key word there, the downhill diffusion of a solid to the uphill transport. So plant cells use this, as far as hydrogen ions go. So here's, let me take that off. So here's our primary active transport, yes, primary. And then secondary active transport is using the hydrogen ions, and kind of like a dam, right? Going from high to low, but we couple it. So this is going from high to low, this is going from low to high, but since we're coupling it, energetically favorable, and we can do the work. That's secondary active transport. Hey, why is this important? Why are we learning this stuff? Well, what we know about co-transport proteins in animal cells has helped us find more effective treatments for diarrhea. Normally, sodium in waste is reabsorbed in the colon, maintaining constant levels in the body. But diarrhea expels waste so rapidly that reabsorption is not possible, and sodium levels fall dramatically, like crazy bad. And so this is life-threatening. Thousands and thousands and thousands of people die from this in developing countries. And so to treat this life-threatening condition, patients are given a solution of drink containing high concentrations of salt and glucose. Why? Because the solutes are taken up by the sodium glucose co-transporters and pass through the cells into the blood. This simple treatment has lowered infant mortality worldwide. Hey, helping people live is great. I'll pause and answer these questions right now, and your notes, okay? So those are the questions that you should answer. So active transport across a membrane. Macromolecules are too large. Well, there's the bell. Wow, that's so loud. All right, I'm going to keep going and try to finish this before chapel. All right, so active transport across a membrane, molecules that are too large or too charged can't pass. And so these things are passed in and out of the cell using vesicles, there's an organelle, via bulk transport. So this is talking about endocytosis or exocytosis. Here's endo. You can see the membrane kind of folding around this particle. Takes energy. Here's exo. Takes energy. And so 3 types of endocytosis brings molecules into the cell. So it's phagocytosis, which is eating, pino, which means to drink, and receptor-mediated, which means you have to bind to a receptor. All 3 causes the membrane to fold, invaginates, it folds around the molecules and forms a vesicle. Okay, so cellular eating. There's a picture of it, if you want to look at that video. It's I think it's some sort of, paramecium eating something, or amoeba maybe. Cellular drinking, its vesicles also form, but they're smaller and bring in fluids and dissolve substances. And then receptor-mediated depends on the receptors. So here's a great picture here. So here's a white blood cell destroying a bacterial cell. That's phagocytosis. Pinocytosis, maybe solutes, smaller, dissolve substances. But here's a target molecule, and the thing about it is you have to have the right not drawing a circle there. You have to have the right receptors there for it to bind. Okay, so an example here is a coated pit appears in the plasma membrane of a developing egg cell covered with layers of protein. So here you can see that receptor mediated. All of it bringing stuff into the cell. Okay, and here's just another example. Exocytosis is materials are leaving the cell and vesicles. Exocytosis is important for the secretion of substances that are made in the cell. And so, for example, pancreas secretes digestive enzymes and insulin. Pituitary cells secrete growth hormones. I mean, we're talking hormones here, and even ligands and various things like that. Human cells use receptor-mediated endocytosis to take in cholesterol. Cholesterol travels in particles called LDLs, low-density lipoproteins. However, that's the bad cholesterol, LDL. And so in the inherited disease, familial hypercholesterolemia, LDL receptor proteins are defective or missing, which causes cholesterol to accumulate in the blood, which leads to the hardening of the arteries, and a risk for a stroke and a heart attack go dramatically up. Okay? And so that's just an example of a practical application of this. So here is a normal cell, LDL receptor, LDL. Here's mild disease, not as many receptors. Severe disease is that, which the cholesterol is just kind of going along. These are just 3 more pictures, and there is that. And here's the last concept check, okay? So hopefully this was helpful. Again, we'll be going over the concept check answers, and I'll be highlighting a couple of things in class. But thank you so much for all your hard work. So proud of you.